coming here. We, of course, have the world's leading experts in the field of AI drug discovery here with us today. I know that this is a much anticipated topic, so without further ado, we'll get started. These iconic pioneers in the field, of course, so well known that they actually don't need an introduction. But I probably need an introduction. My name is Rhie, and I used to be a reporter for CNBC Asia, covering South Korean stocks like LG, Samsung. And I co-founded an MIT startup called Life Canvas Technologies, which specializes in 3D bioimaging and spatial biology. And as I was plugged into this ecosystem, I had this immense itch to really disseminate all the great technology that was happening here left and right. And I wanted to show it and introduce it to the rest of the world. And that's how this panel started. And when I asked all my scientist friends for names and recommendations for this panel, there was the one name that constantly came up. And that was no other than a gentleman sitting right next to me here, Mr. Derek Lowe, often dubbed as the AI reality tech guy. Also democratizing science, because all my CFO friends, administrators, and stay-at-home moms and dads are apparently reading your blog in the pipeline, making it a Sunday ritual, and they're reading about AlphaFold 3. So kudos to you for making that happen.
Derek Lowe. I am probably most well known for In the Pipeline, which has been going for about 22 years now. And if you told me 22 years ago I'd still be writing it, I would not have believed you for a moment. I was sure I was going to run out of material in 6 months. But I've worked at several companies over the years. I'm on my fourth one now, and I'm coming up on my 35th year in the business, working in a lot of different therapeutic areas. But I have not yet put a single drug onto a pharmacy's shelf in the 35 years, and I'd like to emphasize that to people outside the business, to tell them it's not so easy to do that.
There's still time.
No.
All right.
Hi, everyone. I'm Carpenter. I've had my lab at the Broad Institute at Harvard and MIT for 17 years now, focused on machine learning and drug discovery, especially for image analysis. Because I'm rather a skeptic, I've only been ever involved in 2 companies. The first being Recursion 10 years ago, helping them get started. It was based on the research that came out of my lab that was sort of open access software and openly provided protocols. And then now just last year started a cancer therapeutics company called SyzOnc.
It's a pleasure to meet you all. My name is Nwankwo. I am the founder and CEO of 1910 Genetics. We are on a mission to make every pharma company an AI company. And the way we're doing that is by building and embedding an infrastructure layer in which pharma companies can begin, advance, and achieve their transition to AI-enabled discovery, development, and commercialization. We recently did a partnership with Microsoft Corp that's helping to supercharge our efforts to bring this technology internally for pharma companies to fully realize its potential in their digital transformation. It's nice to be here.
Hi, everyone. Great to be here, too. My name is Snyder. I'm the head of R&D at a company called BioMedicines. We use machine learning to make biologics. I am sort of the odd duck in this group, perhaps, in as much as I'm a clinician by training and was in academia where I developed phase 1 stage oncology drugs. And I am a converted AI skeptic, which I'm sure we'll get to later.
Mm-hmm. Excellent. So I had some requests to make this as provocative and fiery as possible, but I'm going to try to keep it civil. And, we will also, of course, talk about the challenges and the barriers and whatnot, but try to end on a high note, optimistic, because all of us here, we need these drugs one way or another, fast. So, Derek, you've been around the longest. I don't mean in Kendall Square.
I know what you mean.
No, in Kendall Square. And you've seen the evolution, right, and history of drug discovery. But ChatGPT is telling me that your view on AI drug discovery, in fact, has changed quite a bit in the recent years. So do you want to comment on that? And I would also like to hear others' views on how AI drug discovery has changed in terms of narrative and perception.
Well, I'd be interested in hearing how ChatGPT thinks I've changed, because as far as I can tell, I've been more or less in the same place. Now, AI has changed, and probably my opinions about it have to do with what state it was in when I was giving the opinion. One thing that hasn't changed is that everyone is extremely enthusiastic about trying to get it to work Some people are so enthusiastic they believe it's already working in the way that they want it to be. But that's been a feature of computational techniques the whole time that I've been in the drug industry. It's very, very easy to get out over your skis with these topics. And the thing is, this is one thing I want to make clear up front. I see no reason, logically, mathematically, there's no reason I can see why we should not be able to do a lot of these things that we want AI and ML techniques to do for us. It's just that these things are very, very difficult. And we've had fits of enthusiasm over the years where hopes get raised and then dashed because we find out how much harder the reality is than we thought, or how inadequate our hardware and software are. You can go back to 1986 and see this famous article that came out in Fortune about the amazing age of computers during drug discovery. It's a hoot. You should look it up and read it. As a couple of guys from Merck looking at a wireframe model of a DNA double helix and what that was supposed to do for drug discovery is clear. But that, even when my old company, Vertex, that I used to work at, when they were founded, that was the Vertex pitch. We are not going to screen and wander around like those big, stupid pharma dinosaurs. We're going to take our computers and our software and just right into the middle of the bullseye with our 1989 computers and our 1989 software. Right, it didn't happen. It couldn't. So I don't know when it does happen, but I don't see any reason why it can't. So I'm what I call a long-term optimist, but a short-term pessimist. If someone walks into my office on Monday and tells me they've got it figured out and they're doing it, I'm keeping my hand on my wallet because I don't believe it. But if someone tells me in X number of years I think we can, then yeah, why not?
I would add that it depends. The term AI in drug discovery is quite ill-defined. We can agree on that. But there's so many different applications within that broad term that they each have their own hype cycles and their own disappointment cycles and so on. And so there's certain applications that are 100% solid, absolutely work. Everybody's using them routinely. I think images are one of the places where that's been really, really panned out very well. So for example, if you want to use an image-based readout for a screen, previously, we would use classical image processing algorithms. Now it is possible. It's not always necessary. It's not always useful, but it's certainly possible now to use deep learning algorithms to identify phenotypes in images and screen 1 million compounds against them and prioritize those compounds accordingly. That's a routine thing that happens all the time. It lets you prioritize good images towards the top of the list, pick compounds to move on. And then there's other ends of the spectrum where it's like, hey, let's use AI to choose targets for a particular disease. Good luck, because there's not a lot of data to train those algorithms. And I guess on the upside, there's not a lot of way to tell that you're doing badly at it. So there's just a whole spectrum. Those are maybe two extremes, but then there's everything else in between, from structure-based design, structure-based predictions, all kinds of different toxicity predictions that are different sorts of applications. So it's a little hard to talk about it collectively.
Well, as with any technology, you'd see a maturation process. And I think that what we're seeing with AI and pharma broadly, and I say AI and pharma to encapsulate the very long value chain of discovery and development. We're seeing a reckoning. The hype cycle did. It was a fever pitch moment. But I think we're at a place where it's measured optimism. We recognize what the barriers are, and those barriers are real, right? Barriers around data, barriers around computing power, barriers around just the sheer reasoning capacity of these models. And so I think it's a good place to be, because it means that now you know what you're up against and you're talking about AI in real tangible terms so that you can actually go knock down these barriers one at a time. And so that's what I'm most excited about where we are today. I'm an in-vivo pharmacologist. So I didn't train as a computational scientist. I didn't care too much for the hype, really. I was on the receiving end of molecules that would be made by medicinal chemists. And I just dreaded being in the vivarium for many days, testing many compounds, more compounds than I cared to test, right? And so I had a vested interest in any kind of technology that would narrow down the number of compounds I had to test. That's all I cared about. In vivo PK studies, in vivo PD studies. And so my skin in the game was more so from someone who had a problem. a real problem that I wanted the technology to solve. But then at the time, I didn't even understand what the tool was supposed to do, or let alone understand the barriers that it was facing. And so where we are as an application of AI, again, is just really, truly understanding what barriers we're facing, being okay talking about it very honestly and openly, and forming the kind of alliances that allow us to hopefully knock down these barriers one at a time. I do believe they'll be knocked down. I think to bet against technology historically just is not a good idea. And so that sort of realism, I think, is where we are. And I think with pharma companies, I think they're also at a place where they recognize that they have a role to play in order to actually see this technology come to fruition. I think the early wave was relying on so-called AI drug discovery startups to figure it out, to do it on their own. And you got pharma companies throwing targets over the wall and disappearing and asking AI companies to do magic and essentially give them molecules that satisfy these long lists of desired chemical characteristics and target product profiles. But I think where we are today is truly a joint effort. No AI drug discovery company is going to magically appear and give you a molecule. Our message to pharma companies is you are the messiah you've been waiting for. There is nobody who's going to solve this for you. And the sooner you start to embrace this technology internally and bring your historic, decades-long capabilities, resources, data, expertise, the greater the chance you'd give it. So I think the companies that will benefit from this technology are the ones who are having the real hard conversations about what these barriers are, eating their own dog food internally and are not looking for some startup to magically wave a wand and everything disappears.
I'll add two quick things. I think one is I come at this from a similar angle to you. When you sign up a patient for a clinical trial, you decide as an institution to take one on, you get what's called an investigator brochure that describes everything that's known about the drug. And to me, having read many, many, many of those, it is completely unsurprising that the success rate of drug development is so low. And so I have a certain desperation about improving that. And I think to then extend that back to what you were saying, Anne, is how do we talk about this in a way that allows us to isolate the spaces that we're trying to impact and measure if we're having that impact? I think there's no reason why AI should be allowed to be hypothesis free. Like, we should be saying, here's the problem we're trying to solve, and here's how we're going to measure if we actually solved it. The way we talk about internally kind of the buckets, each of which has sort of subgroups in it, is to create a successful medicine that helps people. You need to have the right target or targets, create the right drug that modulates those targets in the way you intend, and then do the right clinical experiment with all the operational regulatory pieces that entails. And each of those is having AI applied to it. So I think we'll talk more about how do we then look within each of those buckets to say, okay, here's where the needle is moving.
Yeah, you make a really good point there, because I think that our failure rate in the clinic is the central fact of the entire biopharma industry. It's around 85%, some a little better, some quite a bit worse. And there's no other industry that has that kind of failure rate. Nowhere near. Everything people find inexplicable or disgusting or hateful or whatever about pharma is tied up in that number. So that also means that even though people would love for AI techniques to be magical, incredible breakthroughs next week, they don't have to be to have an impact, because if we've had an 85% failure rate, if we were just to fall on our face 3 out of every 4 times, we would nearly double the number of drugs that get through. And I think that'd make everyone very happy.
You might have to change your nickname here to AI optimist.
Long-term optimist.
Long-term optimist.
At the same time, we have people come in all the time where I work, and probably where you work too, coming in saying, and their slides always start the same, the revolution is here. All of your competitors have signed up. They are ascending into the cloud. You don't want to be left here on the ground.
Crunching away
with your feet in the mud, watching everyone else grow wings. Do you? Do you really? So I'm allergic to that stuff.
Yes, and we've mentioned AI drug discovery, in fact, being inversely proportional as well. And I think that alludes to the fact of target picking being extremely hard still, and human toxicity, anticipating that is still very, very hard. But I think we have a lot of non-scientists here in the crowd as well. So what do you mean inversely proportional, Derek? Do you mean that it's actually harder with AI drug discovery in the state of data here?
No, it's more that the problems that AI is best equipped to deal with in its current state are smaller problems than the ones that we would like to solve.
Would you say those are the problems that we're already reasonably good at?
Or at least we have some handhold on them. We have no handhold at all on predicting human tox, of course, comparatively, or picking targets. I was explaining this to a journalist the other day. I said, you cannot ask an AI, hey, give me a great target for Alzheimer's that no one else is working on. It's not going to happen. So.
Oh, it'll give you a target. I mean, it is going to happen.
It's not going to be right.
But you'll spend $1 billion and 15 years later before you know that
it's right. And we're talking about the data and the quality of the data. I've also had journalists ask me after AlphaFold came out, what's the next AlphaFold? I said, I'm not so sure, because there aren't too many other big piles of data as large and well curated as the PDB sitting around waiting to be machine learning strip mined. We certainly don't have it for target discovery or for human tox. So that's what I mean by inverse, though, is that the things that we're most equipped to attack are things that people would regard as kind of smaller problems, and the bigger and more lucrative and important they get, the tougher it is for AI to work on.
And unfortunately, the more expensive a stage of drug discovery those are, it also seems to be highly correlated, unfortunately.
I think in my bucket concept, the clinical development piece is the space where I think is where there is a quiet, important, incremental advantage that's happening right now. And advantage is the wrong word. Advancement is what I meant. So things like figuring out where the patients are who fit your eligibility criteria. Totally unglamorous, won't get you a new drug. But if it accrues your phase 3 study in 2/3 the speed, suddenly you're starting to shave off time and money. And similarly, regulatory documents, pulling in the data and having the computer give you the first round, even if it only gets it 60% of the way there. Again, that is an important incremental change that when amplified across drug development can make a really big difference. I do agree that the target discovery piece is the hardest piece because of the complexity of human biology. I am known for saying cancer patients die of heterogeneity. These diseases are incredibly heterogeneous. It's not confined to just cancer either, autoimmunity as well.
Oh, gosh.
So I think the target discovery part is, in some respects, the hardest, and in the long run, perhaps the most impactful.
In that holistic pipeline, we're talking about, not just pre-clinical, but we're also talking about clinic to market. Where do we think that AI actually made the most progress?
Yeah, so I would say it's the places where we've had the most data, the most clear inputs and outputs. So protein sequences, protein structures, PDB was full of these, and that seems like a relatively solvable problem. There's less data on chemical structures with proteins, but that is data that exists. There's tons of genome sequences out there. There's genome sequences in mRNA, so linking the two of those is likely to become more and more predictable over time. Predicting protein levels from mRNA levels is a whole nother question, but there's much less protein expression level data as compared. So I think the amounts of data that are out there is really what dictates the ability to input output.
From a molecular level, I'll speak about some of the work that we've done internally. I remember the day that I thought to myself as a pharmacologist, this thing might actually have legs, right? And what happened that day was actually during the pandemic. Like most startups, we had decided to turn our platform to COVID sort of drug discovery. We thought, okay, we know nothing about infectious disease, and how long would it take us to even understand SARS-CoV-2 and come up with something? And there were a lot of efforts at the time focusing on antiviral protease drug discovery. We made the decision to look at host proteases that might be mediating viral entry. And to summarize, one of our AI scientists had done an AI-based virtual screening and generative design of a couple of molecular templates and came up with this structure that the AI was predicting was going to be pretty effective at preventing viral entry. And when presented to a medicinal chemist who was on our team and was a retired Pfizer veteran, and he looked at it and he said, this molecule has no chance at interacting with this target because this target is a serine protease, and this molecule has none of the features that I have come to expect as a medicinal chemist in a small molecule if it will interact with the catalytic triad that mediates the way serine proteases work. And so I'm like, okay, well, here's like really brilliant guy, 35 years in the industry, 1 year experience AI science. I said, well, let's make the molecule. It costs us nothing. And we made the molecule so effective, it cleared the virus out completely. And I looked and I thought to myself, wait, what do we have here? And I think AI is good at reasoning to spaces that if you as a medicinal chemist who's been in the industry for 30-something years, you're fundamentally drawing on patterns, right? You sulfonamides do this. I'm gonna put this ring here and that ring there. And you're walking like SAR encyclopedia. And the encyclopedia in your brain is limited to what you have seen and the patterns that you were used to exploring. What that day taught me was that there is a real limit to human intelligence. And there is a real limit to how much you can memorize. And when you look at that scientist dead in the face and you say, this molecule has no chance because it doesn't look like anything that I'm used to. Well, we did find a new pharmacophore that had no precedent anywhere of ever interacting with a catalytic triad. And that day, I thought, I don't know when this technology will change the industry, but it will. And it will in my lifetime.
Interesting. Yeah, Arthur C. Clarke used to say that if a famous, experienced scientist tells you that something is possible, they're probably right. If a famous, experienced scientist tells you something is totally impossible, they're probably wrong. And you're right about medicinal chemists. We are famously prejudiced about molecules based on what we've been burned by in our career. But there was an experiment done some years ago where a company sent out lists of possible molecules for their chemists to evaluate under the guise of we're thinking of buying these for the screening collection. And what they did was they sent out some of the same ones to different people, and everyone's opinion was different. Everyone's no way we should ever buy this list was different from everyone else's. The most embarrassing part as a medicinal chemist was that they sent out the same lists several months apart to the same people, and they scored them differently. Yeah, we like to sound like oracles, but we're not.
And I think you use the word intelligence, but it's really more knowledge-based. It's really more like, what have you been exposed to? And it's physically impossible for any human to hold all of that in their head. So there's certain things where we really think AI is going to help us out quite a bit because it's just a bit beyond the capability of one human to absorb everything.
Absolutely. I mean, a human being, in theory, could have sat down with the PDB and worked out the pattern matching that is behind AlphaFold, at least AlphaFold 1 and 2. 3 is diffusion, which is a different beast. But you could have in theory, but in practice, I don't think any human would be able to do it, no matter how long you lock them up with a big pile of Mountain Dew to drink.
And then there's things that are just utterly impossible, like finding patterns in mRNA databases that we no human could absorb even one profile, much less a million profiles.
Right.
So at this point, I do want to ask the pragmatic question, when do we actually expect a drug to get to a patient that is powered by AI? And of course, that is a very loosely defined term, but here I mean an AI drug company moving the needle significantly in the entire pipeline. Is it definitely in our lifetime? Probably, yes.
Oh, for sure.
Yeah.
100%.
Yeah, I'm willing
Can you
to
give me
go
a
with
year?
No way. No, I can't, because everyone has a different definition of that, because there are a bunch of people here in this town who will tell you it's happening right now. In fact, it already happened. In fact, they did it last week.
Who are these people?
Just put out a request. You can fill up a panel with them. But the thing is, it's very difficult to come up with new chemical matter. I actually blogged about this today. I don't know if you guys saw this thing, the Belka challenge. This was a little company called Leash Bio that did a huge DNA-encoded library screen, and they had about 300 million data points from their Dell screen on 3 different protein targets.
Which is unprecedentedly
Huge.
insanely large.
It's really,
amazing.
it's the biggest sort of database like that that's ever been made public. And they turned it loose to all comers. And they got about 2,000 entries trying to do machine learning on it and predict binders. No better than chance for new chemical matter or things that the model hadn't seen millions of before. It's very, very hard to do. So again, I don't think it's impossible, but we are going to have to make some breakthroughs before we get a lot of stories like you're talking about the serine protease, where people come in and say, look at this molecule, and I go, I've never seen anything like that in my life. If you want to see molecules like that, you go to natural products, not AI.
Right. Alex?
When you say into patients, do you mean tested in patients or in approval?
Getting it out to a patient so that they can actually take it and get cured.
Approval.
Yes, yes.
I'm going to put my nickel on 2030.
It needs to be in trials now.
As you pointed out, some claim that they are.
I know, yeah, exactly.
Yeah, it really does. It depends. When you say AI, do you mean really data science? When you say discovered by AI, do you mean like assisted by AI? So if we're talking about data science was used in the making of this molecule, then we're already there. If we're talking about like truly magical AI has generated a chemical structure from nothing on the basis of either protein structure or who knows what else, and then it's gone through the whole step, then we're not so close to starting clinical trials on that sort.
I think it's a very important point, and for the sake of this discussion, I'm referring to AI assisted rather than completely novel chemical matter as you referred to.
Yeah, in which case there already are clinical trials started at least, and some failed, I guess.
Yeah.
Jen, do you have a year in mind?
I don't know that I have a year. I think I do know that it doesn't have to be one or the other. I very much view the human feedback that these models are exposed to as being important. And so to continue this story about this molecule that's happened on my team from 4 years ago, it turned out that although that molecule was super potent, it really wasn't drug-like. Right? When you started to pass it through all the battery of, ADME tasks, and you just had all these liabilities and so on. And that's where I saw the opportunity for these to be truly machine augmented, or just the consolidation of like human and machine efforts, right? And then you got your medicinal chemists, your traditional pharmacologist coming back in. I don't think that it's the right question to say AI driven or AI led. I think it's more so there are real opportunities here where AI can help accelerate the work that medicinal chemists are doing, pharmacologists are doing. It doesn't need to be a zero sum. And I think that the more we keep messaging this as a zero sum, it's a lose-lose proposition for everybody. Because in that example, the AI scientists who, designed this novel chemical matter, that was an advance. I consider that an advance. But then now you needed other experts on the team to help sort of think through, well, this is actually not drug-like. And what's that delta between novel chemical matter that is just completely clearing this virus and something that can actually become a drug? And so I don't view this as you win, I lose. And that's been the narrative, and that narrative is destructive.
That's a really good point. This is what Garry Kasparov, former world chess champion, used to call centaur type play in chess. You didn't have all human or all machine. You had humans coming up with these intuitive moves and then turning the software loose on it to see how sound they were working as a team.
Double checking, yeah.
I think another important contrast to lean into is that I think the chemistry field and the biologics field are in different places with very important implications. I won't speak to the chemistry space because I don't work in it. I think in the large molecule space, the roles of toxicity and drug-drug interactions are actually more well established, where if you have specificity, what the likely impact will be in those two realms, which can be very, very helpful. So as we consider what we're looking for from success, I do think we need to think of those two areas separately. Interestingly enough, our first foray was also in the COVID space. And to return to what you said, Rhie, about models, we purposely chose SARS Well, first, because there was a pandemic we thought would help. And second, because the preclinical models are highly translatable to what happens in humans, which is unlike almost any
infectious disease.
And so we went after an epitope that was known to be non-immunogenic, that everyone wanted to go after, but had not been able to using conventional means, and have taken that now all the way through phase 1. It'll be reported out in ID Week shortly. And I think that sort of study where there's translatability and where the translational data from the trial tells you if the drug is safe, effective, well-behaved, and has done something that conventional methods either couldn't do or would have challenges doing, to me, that's how you say, okay, AI support or discovery, whatever term we want to use, did something unique.
I have a question for everyone. I want to know whether we think that drugs that have been enhanced or partly developed by AI, whether we expect them to succeed more frequently in the clinic. Because let's take as a baseline that no matter how you've come up with a drug, there's so many stages towards the end that are like quite old school and routine with, all the admin talks and everything else. So my argument would be by the time you get through all of that, it's pretty much as likely as anything else. Because I'd be very nervous if we were doing AI and then just like spewing predictions into humans. Then I would say, okay, it's much more likely to fail relative to a well-curated human involved step. But that's not what really happens. There's this huge bottleneck of many steps. But I mean, there's also the possibility that they, on average, do a little better. Does anybody expect them to do a little better?
I blogged about that a while ago because Boston Consulting Group put out an article where they talked about the clinical success rates of AI-derived compounds. And I was very skeptical of their paper and I didn't like it very much. And I fielded a call from them about 18 hours later, but they did not want to kill me. I was kind of surprised at that. We had a really civil discussion and they're actually following up on this. But no, I think you're right. I think the problem that the huge problems of target selection and human tox are not affected so much by AI. So I expect at best they'll be about the same for a long time. Eventually, one hopes they'd be better. But I think that they're going to be probably about the same. Maybe, in fact, a little better right now because so many people are picking AI targets and drugs that are very close to what we come up with our own brains. So they've kind of de-risked it a bit. So there may be an artificially high number from that. Yeah.
That is exactly what we're doing. Anyway, I think that for that reason, there may be in some spaces a higher success rate. I also think there are areas where the rules are better known. If you think of oncology, can you target a tumor-specific epitope and avoid normal tissue with specificity? If you can succeed in doing that, then it's quite well known that if you attach some sort of payload to it, whether it's an ADC or a TCE less proven, a CAR, that you'll get a positive effect. So I think it very much depends on the quality of the hypothesis and whether you have preclinical models that actually reflect reasonably reflect what might be expected in humans.
While I do agree that we need new targets, I also think we have enough targets already. We have the targets that are so clearly causative to certain diseases, and we just haven't been able to drug them. Right, so I do think that target discovery is difficult. It's difficult whether you're doing it the good old-fashioned way, like we did in biochem, or you're doing AI, whatever it is. But I just want to make sure that it's sort of pretty obvious that there are a lot of these targets, right, where we're pretty certain the role they play in disease, and just we have not been able to drug them. So maybe we should point AI to those because then those will have a very high degree of success, right? You take tumor suppressor 53. How many pharma companies have been trying to develop a TP53 activator? I think a lot of people are fairly certain that if you could do that, you'll move the needle for certain cancers. But no one has done it. You know what I mean? So there's this target discovery is all well and good, but I think we do have targets that we have not drugged. And so maybe we should start there. And show that AI can, make a real dent for some of these undruggable targets. And I do think, to the degree that we believe in the biological rationale of some of these targets, like TP53, we'll see a better success rate in the clinic. just because they were inherently better targets. Not necessarily because of the AI, but because you appointed the AI as something that you feel incredibly confident about the role that it's playing to drive the biology.
I think that's so insightful that it seems likely there will be a bit of a wave of low-hanging fruit once we crack the here's my protein, give me a compound that does a decent job binding to different aspects of it, which seems to me like one of the problems that will be solved sooner rather than later, just because it is possible to get data on does this compound bind this protein, the Leash Bio being a recent example of a data set. That's a pretty tantalizing idea that there will be kind of bolus of like, okay, everything that we already knew a decent target for, right, let's get through those.
But it's interesting that most companies are not picking those targets, though, right? Those TP53s and those KRASs, people are not picking them. And so I wish more AI companies pick those so that we can collectively move the industry, because it's pretty certain if you've got pharma companies been trying to get a TP53 activator, if you can do it, you're already ahead of the game in terms of probability of success in that phase 2 efficacy study.
Yeah. I can actually take us a little bit out of the target selection space too, because what we were talking about a minute ago was the translatability of the assays. That is something that I'm a real crank about after reading a couple of papers by Jack Scannell over the last few years. He was modeling the whole drug discovery and development process. And at first I was very skeptical because modeling this is kind of a castle in the air type thing. But he convinced me that the biggest factor was the translatability of your assays, especially the early ones. How well do they reflect the human disease? Mm-hmm. And for most of our diseases, the answer is not all that well. That's why infectious disease, our face is all lit up at that, because that actually works pretty well. Diabetes is not bad. Cancer, yeah. CNS, forget it. It's terrible. We don't understand about what's going on in the brain compared to these other things. So if we could get AI to help us out with coming up with more translatable assays, now God knows how you do that. I don't know how to do it. That would be a breakthrough.
Yeah, I don't think you need to come up with new ones. It's the ones we have are sort of medium to low throughput, but if we can use AI to pre-screen compounds and give us 30 good compounds instead of 3 million to search through, then we make them.
You have to be sure it's a good assay, though, because one of Scannell's points that really hit home for me was that he has these graphs on translatability and the number of false positives and false negatives you get. And he shows that at low translatability, there is nothing you can do to get out of that except have a better assay. The temptation is to screen more. Oh gosh, if you can just screen harder, you generate more noise. You cannot screen your way out of a bad assay. All you can do is just dig your own grave.
Yeah, but if you're pre-selecting compounds and you only have, let's say, a dozen core fundamental structures, then you're not scattershotting across the whole chemical space.
I love it that this is getting a little heated here, but switching gears to maybe a different question here. Recursion, where Anne sits as a board, recently acquired Exscientia, of course, and we see a lot of consolidations happening, and as tech is eating the world, it sounds like AI bio is perhaps eating AI chemistry, or this is kind of the elephant in the room question, where is it tech bio or biotech? What's the brave new world gonna look like?
I think data is eating everybody. So I don't know that bio is eating chem, or chem is eating bio. Data is eating everybody. Computer is eating everybody. And I think, again, it's the natural course of events and the maturation of any technology. Consolidation, any technology, it's actually welcome. What he says is we recognize as an opportunity and we're going to join forces to tackle it, right? I talked about barriers around data, around model, around compute. I see it actually as a reinforcement of the opportunity and not anybody eating anybody necessarily. We are all going to find ways to work together. By we, I mean companies in native tech bio, AI drug discovery, pharma companies are going to figure out a different model to partner with these companies, a model that is different from here are some targets, I'm going to walk away and come back and you hand me molecules. That model got to go because it really is just a fallacy that things will work out in that context, right? Pharma companies have got to have more skin in the game. They've got to be more committed to doing a lot of the work to reimagine a lot of how they do discovery internally with AI. Only then would they set up their external AI discovery partners for success, and by definition themselves too for success with some of these partnerships. So I think beyond the recursion exponential merger, there's just a reckoning, just this idea that this stuff is hard, the opportunity is clearly immense, and it's more of a ways of working thing. We're going to figure out what configuration, what way we need to sort of bend, twist, and configure ourselves to function in this new world. So I view it as a natural next step in the evolution of any maturing technology.
Yeah, and I would say it's so hard to predict because it's dependent on the economy, it's dependent on pharma's biz dev strategy of the month or of the year. But I can see certain types of companies working really well in this new world. One is let's do everything really well, which Recursion seems to be going after with Exscientia and Valence and Cyclica before them. Each step of the pathway, let's get the best AI technology for that to create an enormous entity. There's standard big pharma, which will hopefully absorb and internalize different types of technologies along the way. But then what I really want to get to is maybe there's the kind of service provider that does one step really well. And I think that's a pretty exciting mode. Maybe Leash Bio as an example of collecting enough data to do this chemical protein binding prediction really well. But I also expect to see if services become available, it makes it possible for smaller biotechs that have really deep domain knowledge, really awesome translatable assays, depth of target knowledge, they don't have to be experts in all of the AI things. They can interact with some of the service providers to really accelerate things. So I think that's another model that we haven't seen too much of yet.
We're kind of reinventing Greek philosopher Aristarchus. He's the one who said that the fox knows many things and the hedgehog knows one big thing. Isaiah Berlin got a whole book out of that. But I think you're right. You're going to have a lot of different business models for a while, and it's going to be fascinating to watch them struggling to see which one works out. And you probably want me to predict. I'm not going to. I really can't. If you'd asked me back in 1989 when I started in the business what things would look like in 2024, my answer would have been garbage. Now, of course, now I have 35 years more experience, but guess what? My answer now will be experienced garbage.
I mean, I think you all have said it well. We can't really be sure. There's the possibility that large pharma will basically be a phase 3 in commercialization machine, and they'll sort of ingest products. I think we're sort of in that phase to some extent now, although you're trying to change the paradigm, which is admirable and may work, at least in some cases. I agree with what you said, that we're in a phase where there's just lots going on, and there will be consolidation undoubtedly. I also think that whether it's through mergers or collaborations, this need to connect right target, right drug, right clinical development, we will all benefit from all of those things going well. And so some form of connectivity needs to happen for us to realize the potential benefits of these developments.
And I have one final question, and I think I'm going to open up the floor. Derek, you're going to have a new life as an angel investor now, and you're not a co-founder of SyzOnc, not 1910, you don't work for Rhie Bio, but personally, if you had to spend $200,000 on a new AI application, this could be money for your child's college tuition, right? What would it be, Derek?
It would not be large language models. LLMs don't know anything. LLMs say things. They don't know things.
At best, search. Yeah.
I would be fairly conservative about it. I would put it towards some of the smaller, more immediate problems we have where there are decent amounts of data to work with, and we can grind out some better solutions, like we were talking about earlier with finding patients and things like that. I would put it toward that because I think that would be the best near-term use of the money. And I'm 62 and a half. Near-term is where I live.
I think I would say maybe quite the opposite, because I think the companies that are focused on one chain of the methodology, there's going to be a lot of competition for that. And so while I think that they're very likely to be effective because there's good data, I think there's also very likely to be competition and eventually monetization, and not necessarily extreme profits, but rather just like, okay, we can do this. We can predict protein structures. We can do this, we can do that. So I put my money on the smaller companies with the depth of biological knowledge that really understand an area well, but are AI friendly and can kind of pick and choose, 2 years from now, who's got the best generative medicinal chemistry going on, and who's got this and that, and pick and choose from the winners, but staying focused on the ultimate goal, which is getting medicines to patients.
If we agree that AlphaFold 3 or AlphaFold 2 is one of the clearest examples of deep learning and AI working, and part of what contributed to that is the vast amounts of data that Google and DeepMind had available, then I'd be looking to invest in any number of initiatives that help us overcome data scarcity in this space, right? Multimodality being one, I'd be very interested in that. I'd also be interested in investing in initiatives that see pharma companies do more internally. They have the most vast, most relevant data set. I'd be interested in seeing them leverage things like federated learning, for example, to unlock these siloed hidden data sets. So I'd be in favor of anything that breaks down data walls and gets pharma companies. They are going to be the key to this. I don't agree that they can be the phase 3 and commercialization machine and let a bunch of startups fight it out in early discovery. We need their capabilities. We need their expertise. We need them doing this internally. We need every pharma company to be an AI company. I will invest in that.
I'm assuming, for the sake of this argument, that I'm making a long-term investment. A little bit of contrast to Derek. I think there are some great companies and academics working in the spaces of the clinical development problems and the making the best drug problems, including hopefully yours and mine. I think where the greatest data scarcity that can lead to the next wave of transformation is actually generating data on heterogeneity and the attendant assays. I think we're in the low-hanging fruit phase, and I want to think ahead to, okay, what's next? When can we really know exactly what drug a patient needs, rather than this cycling through things with a pretty good degree of randomness, which is the current method in many disease states.
So I think everyone got their share of secret tips to becoming the next billionaire. So thank you so much for your insights. And now I want to open up the floor. Let's get 1 or 2 questions.
The paper says that AlphaFold 3 works better, but I can't verify that because none of us in pharma can use AlphaFold 3 unless we sneak on to the public face of it on the weekends from our home IP addresses, and no one's doing that. But I think that it actually is working better, but the whole diffusion modeling idea, frankly, gives me the willies a bit, because I think the way some people have it pictured is almost violating a sort of conservation law of knowledge, that you cannot distill knowledge out of thin air. And what we're doing is we are, again, leveraging the things that we know about, the way things bind and the way we know about them, and trying to make stuff that looks like that. And the diffusion method, to me, is a very efficient way of doing that, of finding stuff that looks like what we already know about. And I am not an expert in this. Diffusion techniques are just an extremely good way of doing that, but I don't see them as a breakthrough toward generating something new. In fact, they're quite unlikely to generate something that's radically new.
Just like the LLMs, really, that at best they kind of search in a way that humans can't possibly search the entire internet, but they can find things that make sense.
Exactly, or they don't make sense because they don't know what sense is. They find things that look like other stuff that people have said. And as the internet fills up more and more with AI-generated text, these models are going to be breathing their own fumes, and that's going to be a problem.
I'll say 2 things. First is narrative, and second is incentive. I think the narrative as to, AI is going to solve all the problems. It's going to eliminate you as a scientist. We don't need you. No one wants that, right? So I think a lot of the repulsion, if for lack of a better word, has just been the way it's been messaged. And I hope that with a little bit of this moment of reckoning that we're having, it's messaged more as a counterpart to what you do, unless it's something that's here to replace you. And I think the second is around incentives. If you think about the way people are compensated in pharma, just think about the compensation structure. And people are not incentivized to innovate. They're incentivized to get you a molecule. And so if you're not going to incentivize me or acknowledge that I might have spent the most part of my year this year in really trying to be a good counterpart to my AI colleagues and provide the substrate for them to experiment, if at the end of the year you're going to write my performance appraisal just based on did I get a molecule or not, like what incentive do I have to be a good collaborator with the AI people? And the AI people keep getting chopped in the back in some of these digital groups that nobody really thinks about, and so on. So I think the way it's messaged, the narrative and the incentives of these R&D groups, those two things need to change. And then you'd see people being less put off by some of these. And then, everybody likes innovation, but no one wants change, right? And so you have to make it from a ways of working perspective, assuming you get the narrative and incentives right, you just got to meet people where they are at, right? Make this as user-friendly as possible. And so those are the three levers I'd pull to see more adoption of some of these tools within pharma.
I think these are such important questions because ultimately we must work with the regulators to get these drugs to patients. So I'll take that in two pieces. I think the AI-based drug development piece, because of the studies that are required to get to an IND, ultimately it doesn't really matter where the drug started because that set of studies is fairly standard. As you referenced, you can't just leap, and nor should you, leap from the computer into the human. We've interacted with regulators in 3 different countries and on 2 different molecules, and we've had no problems with any of them with respect to the AI piece because I believe of the fairly conventional IND package. I think in the toxicity space, gosh, we would all love not to have to use large animals, well, any animals, but especially not the ones that resemble us or our pets. But the bar is very high because if not them, us, right? And so I think the FDA is trying to motivate that change, but until we have the translatability from the multicellular system or whatever the predictive technology is all the way through to the humans several times, I just think it's going to take a long time to get there. I do think the FDA really means it, but I think the burden of proof is high.
Okay. Unfortunately, that is all we have time for today. Everybody, please give a round of warm applause to this exceptional panel. Woo. Thank you so much.